Now friends, after seeing that what is plasma membrane and what is its significance, seeing its versatile nature and its role performed in the different functions of the cell, it is very necessary to understand the structure of or you can say architecture of the plasma membrane. Architecture of the plasma membrane means how this plasma membrane, what is the structure of the plasma membrane, what is the chemical composition of the plasma membrane. But one thing is, as I told you in the in my previous video, that this plasma membrane is very thin in diameter. As a result, it cannot be observed through the general microscopes which were available at that time. So, whatever the information which we get about the plasma membrane in 19th century was by its chemical analysis. The first person who proved that there is something like membrane around the cell present was Overton. Overton, he proclaimed in 1899 that the plasma membrane has the property of allowing certain substances to enter inside the cell, and the substances which can enter easily inside the cell were all non-polar in nature. Non-polar in nature. For example, ethanol, oxygen, carbon dioxide, benzene, means the substances which are soluble in lipid can easily pass through this membrane, and the substances which are polar in nature like sodium ions, potassium ions, calcium ions, they cannot enter inside the cell. On that basis, Overton said that there is something present around the cell which is called membrane, and this membrane is chemically probably made up of a lipid layer, and that lipid molecule may be lecithin and cholesterol molecules. Till 1890 we have only information that this is made of lipid molecules, but in 1905 Langmuir in 1905 said that the membrane which is present around the cell is made of a monolayer of phospholipid molecules. This is the monolayer model. Monolayer model. He proposed a monolayer model. These are the phospholipid molecules which forms a single layer around the cell. But later it was found that the amount of lipid present in the membrane is much more than to form a single layer around the cell. So, Garter and Grendel in 1925 said that it is not made up of a monolayer of phospholipid molecules, but in fact it is made up of bilayer This model is termed as bilayer model. Since it is made up of lipid, so it is termed as lipid bilayer model. So a similar arrangement of phospholipid molecules was present on the other side. So in this way, two monolayers cover the whole cell. This is one layer and this is another layer of phospholipid molecules. These are the hydrophilic ends in fact, hydrophilic ends of phospholipid molecule, and this is the hydrophobic layer. Hydrophobic layer. But soon it was realized that enough lipid molecules are not present to form the two complete layers around the cell. Secondly, the surface tension which was detected around the cell was much less compared to the membrane, which if it is made up of only two layers of the phospholipid molecules. मीन जो surface tension नापा गया प्लाज्मा मेम्ब्रेन के सराउंडिंग में वो काफ़ी कम था, जो कि एक दो लेयर की वजह से सरफेस टेंशन होना चाहिए था. और soon the scientists realized that this is not made up of two complete layers of phospholipid molecules, but in fact protein molecules are also present in the phospholipid bilayer. So on that basis, Debson and Danieli proposed another model which is termed as sandwich model. Debson Danieli in 1935 proposed the sandwich model. Sandwich model to explain the structure of plasma membrane. According to Debson and Danieli, the phospholipid bilayer forms a two-layered wall around the plasma membrane, and the protein molecules are present on both the outer surfaces of the phospholipid bilayer in this fashion. These are the two protein molecules, protein layers, which are present both towards the outer face and inner face of the plasma membrane, means towards the cytoplasm and towards the exoplasmic phase. So since this structure was just like that two breads, in between the two breads something is put, that is the phospholipid molecule. That's why this model is termed as sandwich model, which was presented by Debson and Danieli. So in this way, till now we came to know that the layer which surrounds the membrane is made up of two things, that is phospholipid molecules and proteins. But soon it was realized that this may not be the arrangement, because first thing, that these phospholipid molecules were not enough to form the whole complete phospholipid bilayer, and also it was found that there are a number of proteins which were hydrophobic in nature, and they were present in between the phospholipid bilayer. Thirdly, an enzyme, that is phospholipase, was used. Phospholipase, as the name indicates, phospholipase means it dissolves, it digests the phospholipid molecules. When this phospholipase enzyme was applied on the membrane, then it was found that this phospholipase enzyme was able to dissolve these phospholipid molecules, to digest these phospholipid molecules. This was only possible when this प्रोटीन लेयर वॉज एब्सेंट, क्योंकि यदि प्रोटीन लेयर प्रेजेंट होता तो ये फॉस्फोलाइपेज एंजाइम को अंदर नहीं जाने देता इसको डाइजेस्ट करने के लिए, लेकिन चूंकि फॉस्फोलाइपेज एंजाइम अंदर पहुंच जा रहा है मींस कि ये प्रोटीन लेयर कंटिन्यूअस नहीं है बल्कि जगह जगह से टूटा हुआ है. So on that basis this Dawson-Danelli model was also discarded. Then in 1950s, when the electron microscope was discovered, at that time another model was proposed by Robertson to explain the structure of plasma membrane. Robertson in 1950 he proposed a trilaminar model. Actually he took the photograph of the plasma membrane by electron microscope and he saw that this photograph shows 3 layers around the membrane. One layer, second layer. These were the 2 layers which were present, which were seen in the electron micrograph. Outer layers. They were very dark and the middle layer, this one, was light in color compared to 2 darker zones which are present towards the outside, towards the periphery. The thickness of this membrane, he proposed that it is about 7 to 8 nanometers. On that basis, he said that plasma membrane is a trilaminar structure, 2 darker zones and light middle zone. The light middle zone is about 3.5 nanometers. This is 3.5 nanometers. While the 2 darker zones, 2 outer layers are made of 2 nanometers each. This is 2 nanometer and this is also of 2 nanometers. And he said the most important part of his model was that this type of membrane is present in all the structures, whether it is plasma membrane or the membranes which surrounds the intracellular organelles like nuclear membrane, mitochondrial membrane, endoplasmic reticulum membrane, lysosomal membrane. All the membranes are made of same nature, and on this basis he termed this model as universal. Means it is everywhere present. This structure is present in all types of membranes, or you can say biomembranes. The universality of this model was most important thing, but it could also not able to explain all the functions, all the activities which takes place inside the plasma membrane. So so far the most newest model, which is in fact the most accepted model, is fluid mosaic model, which came into existence in 1972 and which you will see in my next video. So because that needs certain long explanation, so you watch the next video to understand that how fluid mosaic model presented by Singer and Nicholson was able to explain the architecture of plasma membrane. Okay, thank you.